Okay, welcome back to the Classics of Immunology Journal Club, a la Kendall, that we're doing at least one paper every week, and we started in 1798, and now we're up to 1983. But before we get started on this particular paper, don't forget to like the video on the YouTube channel, and then click on the bell, and then also scroll down and check out my website, and particularly looking on the writing tab, you will find all of the classic papers of immunology listed there. So today we are going to review probably one of the most earth-shaking papers of the entire 20th century in immunology. It's a new paper that was published in February of 1983 by Stefan Meuer, Kathleen Fitzgerald, Rebecca Hussey, James Hodgdon, and Stuart Schlossman, and Ellis Reinherz, who was the senior author. And this is out of Ellis's lab as a new young investigator. Now, the title of this paper is Clonotypic Structures Involved in Antigen-Specific Human T-Cell Function, Relationship to the T3 Molecular Complex. And the subtitle on the subsequent pages is particularly revealing because it's the human T-cell receptors for antigen. On the background of the workers in this paper, it's the same three players in the paper that we did last week or the week before, except with the addition of three technicians, Kathleen Fitzgerald, Rebecca Hussey, and James Hodgdon. In the introduction of this paper, the authors go through the background that led up to this paper, and they first review their previous publications, and they had a whole string of them going back to 1978, 79. So first they reviewed their papers on the T3 molecular complex on the surface of all T cells, so that it was a pan T cell marker, that they discovered antibodies to the T3 molecule would block both proliferation and the generation of cytotoxic T cells. And so therefore they thought it had something to do with antigen recognition. It would block antigen stimulated proliferation and the generation of cytotoxic T cells. Next, then they reviewed all their work on the T4 surface molecule, which is now called CD4, and the T8 surface molecule, that's now called CD8. And as you probably already know, T4 marks about 60% of the peripheral T cells, mature T cells, and T8 marks the other reciprocal 30-40%. And they had shown that antibodies to monoclonal antibodies to these two surface structures would block the generation of cytotoxic T cells, but not proliferation. So that was a little bit strange. But they went further in the last paper that we reviewed and showed that these surface structures were really responsible for what came to be known as MHC restriction of the T cells. They could only recognize antigen in the context of either class 1 for CD8 T cells or class 2 for CD4 T cells. And that was very, very important. So they assumed, or they concluded from their experiments, that these two surface molecules had something to do with antigen recognition, but they weren't sure exactly what. But they went on to say then that these experiments led them to the hypothesis that there had to be a clone-specific surface structure that was specific for one T cell. It was very much like the antibody molecules on the surface of B cells that were clone specific, that would lead to antigen recognition and then activation of the immune response. And so therefore they decided that what they needed to do is to identify whatever this stuff was that they needed to make T cell clones, which they had done and we reviewed before, and then use one of them or other ones as well, to immunize mice to see whether they could get monoclonal antibodies that would recognize the T cell antigen receptor. And I just wanted to say parenthetically at this point, it was clear from our experiments that we published in January of 1979, so this is like 4 years previously, when we said it was possible to generate antigen-specific functional T cell clones, cytotoxic T cell clones, it was clear to everyone in the T cell business that this was the way to get at the T cell receptor. You make a T cell clone, antigen specific, and then you immunize mice, get monoclonal antibodies, and then you're off to the races. So they did that. They say in the introduction that they got 2 antibodies that would, from the immunization of a single T cell clone, that would block both proliferation and the generation of cytotoxic T cells. And these antibodies recognized surface molecules that were different from what T3 recognized and what T4 recognized and T8 recognized. So in the materials and methods of this paper, for the lymphocyte populations, they used just normal peripheral blood mononuclear cells that they got from high-peg density gradient centrifugation from normal people. And they used, for stimulators in their mixed lymphocyte cultures, Epstein-Barr virus positive long-term B cell lines. And one in particular was called LAS-156. In order to generate IL-2, because you couldn't just buy it at the store like you do today, they used normal PBMCs, activated in vitro with PHA and PMA, 4-ball myristic acetate, and they threw in the LAS-156 cells to see if they could hop up the system by getting them an allogeneic stimulus. They'd already made human T cell clones, and one of the things that Ellis told me subsequently is that he was the donor for all of these T cell clones, and the stimulator cells in a mixed lymphocyte culture that generated cytotoxic T cells was this LAS-156 B cell line. And they cloned them out using limiting dilution with 5% conditioned medium that contained IL-2. And they selected, for the experiments that they're going to talk about in this paper, one clone that they called CT83, and I'm just going to talk about it as clone 3. They had other clones. They had a whole bunch of other clones, both CD4s as well as CD8s. So in order to use this number 3 clone, in order to generate their monoclonal antibodies, they immunized mice with 5 million cells intraperitoneally. And then after 10 days, they boosted these mice, the same mice, with the same stimulator clone cells, number 3. And they gave their second immunization, their booster immunization, both IV and intraperitoneally. And then 3 days after that booster shot, they gave them another dose, both IV and IP. And then they cloned out these cells, 96 well plates, by limiting dilution again. And after 2 weeks, they had hybridoma cells growing in about greater than 200 different wells. And those were the supernatants from these wells, were what they then tested to see if any of these hybridomas were making antibodies to the surface of clone number 3, and were specific for clone number 3 and wouldn't react with anything else. That was critical. After they thought they generated some antibodies, they needed to characterize these antibodies. And so they did a series of experiments looking for just expression of the surface marker on the cell surface. And I'll show you some of those data. And then they did experiments where they tried to modulate the different cell surface markers, T3, T4, T8, and the clonotypic. They called their antibodies clonotypic or clonotypes, as a takeoff from isotypes, which are the antibodies, the B cell antibodies. So they also did competitive inhibition of both the generation of cytotoxic T cells and also the generation of antigen-specific proliferation. And finally, they did some biochemical characterization of the surface molecules on the clone 3 that these antibodies recognized, these 2 different antibodies that they generated. And what they did was they iodinated the cell surface with I-131 with the enzyme lactoperoxidase, and then they tried to immunoprecipitate from the lysate from these cells whatever these antibodies reacted to. So in the results of these experiments, and now we want to do some screen sharing. So on the left on this screen, you can see figure 1, and this is just simply a flow cytometry plot against two different clones. This is clone 3, this is clone 4, which happened to be a CD4 positive cytotoxic T cell clone. In the gray areas here is the staining of anti-T3. So they're both T3 positive, both of these clones. But if we look at the clone number 4 first, the specific antibodies, the clonotypes A and B, were negative on this negative T cell target, whereas they were clearly positive on the clone 3. And then if we look over here to figure 2, and we've got plotted here the percent specific lysis, we're looking to see whether or not pre-incubation with any of the antibodies would block the generation of cytotoxic T cells. And so this is our control, whether it's just medium alone, and we've got a 50% lysis here. And they used a 20-to-1 effector to target cell ratio. This is anti-T3, and it clearly brings it down. This is the A antibody clonotype, and this is the B. Yeah, this is anti-T8, and this is another T8 that's negative, and this is T12, which is a pan T cell marker that does not block immunologic activity. I am going to then go to the last figure in this paper, where they surface iodinated these cells and then immunoprecipitated them with cephalose-bound antibody. And this is the A antibody, this is the B antibody on to clone 3. And you can see that there are two bands here, one at 49 KD and one at 43 KD, that both of these antibodies bring down. And these gels were done under reducing conditions with 2-mercaptoethanol. So if you didn't do that, you got a single band at about 90 KD. So I'm going to, now you can, if you go back and read through this paper carefully, you can go through some of the other experiments that are proving their point, but I didn't think it would be, we might get into the weeds if I did them all here. So those were the data. And so clearly they had something that was earth-shaking that nobody else had before this paper was published. So in the discussion, they go through all the points that they've made from the data that were in this paper. Number one, that they've raised clone-specific monoclonal antibodies. And these antibodies block both proliferation and the generation of cytotoxic T cells. Furthermore, these antibodies recognize something on the T cell surface that's associated closely with the T3 molecules, but they're separate structures. Part of that, they say, is because the T3 brings down a 20 KD complex, whereas the clone-specific are bringing down 49 and 43 KDA. They say it's in the discussion that it's likely that the clonotypic antibodies recognize something that's variable on the cell surface from one T cell to another, whereas the T3 molecule, the T3 antibodies that recognize T3 seem to be recognize something that's constant that all T cells have. So it's like the constant region of the antibody molecules. They go on to say then that the T4 and T8 molecules are precipitated. They precipitate down to 62 KDA and 76 KDA. The final point that they make then is that whatever these antibodies are recognizing, it seems to be mimicking what antigen will recognize, what is recognized by T cells, and that is that it triggers the production of interleukin 2 and therefore proliferation and therefore the generation of cytotoxic T cells. So that's it. And subsequently, I just want to point out several things about this paper. As I said earlier, people in the field that were really interested, the nature of the T cell antigen receptor was unknown for at least a decade when T cells were first marked in around 1970. So everybody in the business wanted to be the ones that did this because they recognized that this was hopefully going to lead to a Nobel Prize someday. This group, led by Reinherz, published 4 more papers in 1983 on the nature of this T cell antigen receptor. They had a paper, so their first paper came out in February in the Journal of Experimental Medicine. In June, they had a paper in Nature. And then in September and October, papers in the Journal of Experimental Medicine again, and then another paper in October at Cell. And when I read through these papers, I said, well, how do these people have any time to write these things and still do experiments, for God's sakes, you know? But anyway, you know, there other people were breathing down their necks. And so I understand the anxiety that they must have been going through at the time. And in particular, there was a group in Colorado that was led by a man and wife team, John Kapler and Pippa Merrick. They were working in the mouse. Now, to raise monoclonal antibodies in the mouse against mouse cells or mouse molecules was a heck of a lot more difficult than immunizing mice with human T cells, human T cell clones and so forth. Everybody knew that you needed to get monoclonal antibodies to human T or to T cell clones. And since Merrick and Kapler were working in the mouse, they were distinctly disadvantaged. In addition, the mouse T3 molecule had not been discovered at that time. Neither had this T4 or the T8. Now, it turns out that there were surface structures that they thought recognized specifically the helper population, and it was called LYT1. And then the killer population was called LYT2. And it turns out that LYT1 did not recognize CD4, but LYT2 did recognize the mouse equivalent of CD8. So they didn't have the whole picture, not like Reinherz did, and not with the lead-in with at least a dozen papers that were leading up to this particular paper that was published in February of 1983. I read through all these papers and I said to myself, these guys, Reinherz and his crew, were the first, because it was February of 83. They were complete because they had the whole ball of wax. They had the signaling molecules, CD3, T3, and they had the associative recognition structures of T4 and T8 that were responsible for MHC restriction of antigen. And they were correct. They didn't have to go back and revise because they didn't interpret this right or that right or whatever. So my take on this paper is that it's probably one of the most stellar papers of the whole century and the 20th century. And it deserves recognition by the world because everything that followed thereafter in immunology was initiated by this paper. So I'll stop there. Don't forget, like this video if you liked it. Click on the bell notified for some more, and check out my website and all of the classic papers. So this has been fun. Thank you so very much.